So I guess we will open it up for questions. You can either post a question in the chat or you can unmute and ask it.
Oh, I'm really fascinating. Thank you for this wonderful presentation and amazing. So did I get that right? There is a vacuole within the vacuole.
Yeah, or vesicles within the vacuole. I guess
So there might be not
defining those terms maybe maybe is something that we should work on if we call the central one a vacuole, whereas the smaller peripheral ones are vesicles. I guess we could probably hash out that
But
terminology.
they are also in a vacuole because usually the vacuole is surrounded by a membrane. So you have a very thin layer outside, which is actually more like a network we've seen already in the scanning electron micrographs. But then you have these vesicles which are actually with cell wall. So technically that are in the vacuole, which is surrounded by a membrane. Is that right? Did I get that right? Technically for the cell, if it's surrounded by an invaginated cell wall, that would be outside actually.
I was going to say some of Dalton's images, especially the ones later on there, they have this region that is, a vacuole shaped region, but that has like a different pH or whatever. But that seems smaller than what you kind of think of as the canonical like vacuole that extends all the way to the edge.
Mm-hmm.
But some of those do have like a membrane bound Dalton, like there's the Bodipi labels that, right? That margin. So that's membrane bound.
It's different though too, right? Like we have different Thiomargaritas. When you're talking about the difference between like Nelsonia-like cells and these elongated football shapes, and then you have Namibiensis cells where you have these large, massive cells. Yeah, the I would say if we're calling it the vacuole, is it's obviously much smaller, and there's a much more cytoplasmic region, I would say, on average in Nelsonii cells, whereas in Namibiensis cells, the cytoplasmic region is much smaller.
Yeah, that's the other complication, that we actually might have a whole bunch of different bacteria that are slightly different. But I wonder, do you think that the Thiomargarita can control these endobionts? Or that maybe this invaccination or this putting them into a vacuole might be the control?
I mean, just based on the kind of these bifidobacterium results, it seems that there is proton pumping into that central vacuole, which is, to my understanding, pretty similar how like lysosomes would work, where you have acidification of foreign particles to try and get rid of them. But the interesting thing would be, and we're kind of in the process of doing it, is if there is nitrate in there, and maybe that's favorable for the endobionts, and it's a way of tricking Thiomargarita and saying, "Hey, bring me in there," and I'll take that.
Yeah, I'm going to chime in and say that we do know, and we just haven't gotten good images of it yet with fish, that the endobionts do collect on the outside of the cell at the division planes. So I've seen like collars around, the division plane, and I am assuming that's on the outside. I haven't gotten good enough images to say for sure, but there definitely seem to be attracted to Thiomargarita, and there's more than one. It's not like a single group. It's like there's a clade, but even within a single metagenome, we're seeing more than one of the same group. So they're attracted to Thiomargarita, whether Thiomargarita is doing something to attract them or that they're just naturally attracted to them. But these are groups of bugs that are very good at symbiosis, right? And they know how to infect a cell, to get inside of a cell. And these secretion systems are probably part of that. So it's complicated, but there's definitely signs of horizontal gene transfer that's between these clades and the Vegetoaceae. So there may be a long relationship there.
Yeah, these secretion systems, they have the smell of parasites. I agree. That doesn't quite fit into the overall picture. But what if the Thiomargarita attract them, then engulf them, and then let them work a while with sulfate and nitrate, and if they're not don't have enough sulfite for them, they just lyse them.
Yeah, it's entirely possible. We just don't know. You know, without a controlled system, without being able to culture Thiomargarita, it makes it very difficult to get the history of what's happening.
Well, from an evolutionary standpoint, those bacteria inside the endomicrobiome there must have a reason for being there. Right? But in your case, I don't see that there's a particular group of organisms that would even help the host cell, anything that the host cell couldn't do by itself anyway. Is that right or wrong?
It's hard to say because, you know, if you think about inorganic nitrogen and sulfur utilization, there are so many different pathways within there. And it could be that there's an exchange of intermediates that somehow or another allow this to persist.
Mm-hmm.
But it's very hard to say. No, there's not, you know, for example, nitrate, there's no stain for nitrate. How do we track nitrate in a cell? It makes it kind of challenging to figure out what exactly they're doing. But what we can say is that, you know, Dalton's now working with cells that are almost 2 years old sitting in sediment. And even when we're trying not to get endobionts, he seems to be getting them. So,
Yeah.
is it just because they're stressed and they seem to be taken up more? I just don't think we just don't know.
Well, and on that note too, Bev, it's interesting though too that their Thiomargarita is also still surviving after a couple of years, like in our refrigerators with an infection, right? Because we're showing that they're actively incorporating all of this stuff, so they're still persisting, you know, maybe in not the most favorable conditions.
Excuse me, have you ever seen them divide inside the Thiomargarita?
Ooh.
Well, we see those you know, groupings of endobionts across the division plane in the Thiomargarita cell that's dividing.
To do that kind of
asking about the endobionts replicating inside, right?
That would be a difficult experiment to do because Thiomargarita doesn't really like oxygen a whole lot. And so if you really wanted to try to do some sort of time-lapsed experiment that might last a week or so, it would be challenging to keep Thiomargarita healthy enough to do it.
Yeah, well, I do not see them accumulating either towards the outside where they had more access to the oxygen, nor to the vacuole where they would have more access to the nitrate. They seem to be all over. I can't see a chemotactic attraction of any of these organisms to an environment that would benefit them.
And yet they are, I mean, I can see, I've seen just giant collars of them around Thiomargarita that's dividing. So they're attracted to something.
I mean, it could be the nitrate. If they can't accumulate nitrate and there's no nitrate in the exterior environment or low, you know, then if they get in there, they have access to electron acceptor, right?
But then I would expect them to accumulate around the vacuole.
Yeah. Well, we know they're actually, you know,
yeah,
they're metabolizing because they're taking up the amino acids. So actively dividing. I mean, I don't know that we have shown that. We have not shown that. Oh, actually, the TEM, no, that's not true. The TEM
Yeah, that's true. I was going to say that too.
Shows some endobionts in the process of dividing.
And then you say also that some are motile, right? So that means they have flagella. Have you seen that?
Never seen any indication of that kind of motility ever inside of a cell. It's Brownian motion.
But if they migrate towards Thiomargarita, they must be able to.
But that's the case with the sulfur oxidizers endosymbionts of animals as well. They stop expressing their flagella once they're inside.
Other questions? You know, I like your endosymbiosis theory, that this could be a step towards the development of a kind of an organelle within a bacterium. Would be great, wouldn't it? But then you would have to find a real function for that endosymbiont, which I cannot see at the moment, or can you see one? Which endosymbiont would be most likely be turned in an endosymbiotic organelle of bacterial origin?
Yeah, it's a good question.
Yeah.
I mean, the way I kind of look at it is like there are different kind of evolutionary, trajectories, and that like the one that led to eukaryogenesis had required certain characteristics, right? And this Thiomargarita system has some interesting kind of similar characteristics, but it doesn't mean that it's necessarily going to lead to an organelle per se. But that doesn't mean that it's not, interesting to kind of look at it for its own sake and for what it might tell us about kind of the evolutionary trajectory that leads to organelles.
And I think, and Dalton didn't cover this, but we have shown that there's outer membrane inside of those vesicles as well. So this is very similar in some respects to phagocytosis. One of the things we're looking on is the mechanisms of this, gene-wise and whatnot, like actins and dynamins and stuff like that. Go ahead.
What I don't understand with the sheaths, how does the sheath work out in this scenario? If they attract bacteria, engulf them, and then digest them at some point, why having a sheath around that keeps them away from the cell actually? Or
that's an extremely good question, because when I was also working on, because there's basically the other paper I was working on is that the ITAG sequences, and we found 123 different clades that are, or species that are statistically significant associated with Thiomargarita, and many of those are known to not be able to live on their own, like nanoarchaea. But then you go, well, what if that sheath? I mean, I could see like a Bacteroides eating the sheath, but how are an organism like a nanoarchaea surviving with Thiomargarita and getting through that sheath? Because it seems like they would have to, right, in order to co-metabolize with Thiomargarita. It's baffling. It's absolutely baffling me. I don't understand how they're getting through that sheath, and that's something we need to look at very closely.
And that they are food for the Thiomargarita, that they are being digested by the Thiomargarita.
We just don't know.
Yeah.
It seems feasible, because I think Dalton's showing that the vesicles kind of seem to like they bleb off and they kind of travel and maybe join up with the giant central vacuole. And so in theory, you could say that sounds like a lysozyme, but to actually track a single cell and follow it through Thiomargarita would be extraordinarily challenging to actually show that that's happening. Yeah, if people have ideas of how to do these experiments to long enough that we could follow something like that. But an organism like Thiomargarita really doesn't like oxygen. And we have a limited time span. Once you pull it out and you start trying to do your experiments, to where it's becomes unhealthy for it.
Mm-hmm. There are one question on the chat by Natalie Elizabeth. Nice talk. What was the fish probe specific for in the fish image? Who are the endosymbionts? Probably the ones that are tagged by fish.
Do you guys want me to answer that?
Go ahead.
I designed the fish probes off of several metagenomes that we did that had endobionts in them. There's really kind of two clades that I designed them off of. They're both gamma proteobacteria and they're closely related. One is more closely related to the endosymbionts of the tube worms, and the other one's more of an environmental clade. So they're pretty specific. And when I did a search on the Silva database, it only hit a couple other gamma proteobacteria in the entire database. That being said, now that I'm going back through the metagenomes again, I'm finding that the tube worm symbionts are in all my metagenomes. And so I don't know for sure if that other clade is even endosymbionts at this point, but they are statistically significantly associated with Thiomargarita from our other study. So they're pretty specific to those two worms. So far, every time we've used our fish probes, we've always found them inside of the cell and didn't see much indication of other bacteria or archaea being inside of the cell.
Thank you. Then there's one by Barbara McGregor. That's for Dalton. In the 3D reconstruction, it seemed that the red shapes were of different sizes and shapes. Did any seem to be dividing? If they were, a single type might expect a single size.
Let me share my screen. I think I have it right. Well, this would be here on the sheet. But one possibility in terms of like the inside is that if they are hitting the endobionts, you're also getting movement in some of them. So capturing, a 400 micron stack, you're going to get movement in there, which kind of distorts some of the actual cellular sizes that you would see. So if you're seeing small differences, it could partly be due to that too.
Mm-hmm.
But if you're talking about out here, this is outside on the sheath where there's a multitude of different microbes out there.
But Barbara had in mind the more cartoon-like picture. That was
yeah, the one I showed. Yeah, so that's I think a maximum intensity projection of the fish. This one.
Yeah, that's the maximum intensity.
Yeah, and you're right. There are some size variation there, and I don't fully understand why that is. We don't see that with the like TEM, for example, and we don't see that with a lot of like the fish. But here, I think
it's just a big cluster of endobionts, and because this is like it's a computer graphic rendition, it just smooths it out. That's not a real smooth thing.
Yeah.
But
yeah.
Our size is real.
The size is real, but it's not this smooth thing. It's a cluster. It's probably a cluster and it's fluorescent, so you don't really see the definition. This is one of the biggest challenges that we run into, is that to get a good image of Thiomargarita, you can't get a very good image of the endobiont. To get a good picture of the endobiont, you can't get a good picture of Thiomargarita.
Yeah.
it's hard.
Yeah, and I think if there's varying intensity differences with when you're doing those max intensity projections, I think that's going to change the smoothing factor that it has and the overall size. Even if there were the same amount of cells there, but the intensity was different from one to the next, that could also influence that.
Okay, let's come slowly to an end. And I would like to ask one more question to Dalton. I have a beautiful picture of this Thiomargarita on the seminar website that you made with the epifluorescent microscope with the green filter, and they are full of yellow particles. Can you tell me, what do Thiomargarita actually store in the cytoplasm besides the vacuoles, in the cytoplasm?
In the cytoplasm?
Yeah. I guess these large particles are elemental sulfur.
Yeah, that's what I would assume. I can't recall which image you're talking about. If they're smaller though, and they're acidic, they could potentially be polyphosphate too, if that's being hydrolyzed.
Yeah, that's what I would want to know. Sulfur, polyphosphate, glycogen, are those the ones that they can store?
Yeah.
The glycogen too.
Yes, I have stained for glycogen. Not PHA, but glycogen actually.
Okay, so that's an indication that they can ferment. Right? They must break down that glycogen.
Good point.
No, it just gives such an enormous versatility of metabolism that you have in here, by the Thiomargarita itself, and then in combination with all the endosymbionts. not to talk about the epibionts that might deliver and draw things away to make metabolism inside the cell thermodynamically more favorable. So an extremely kind of a little ecosystem in itself. And you have found so many hints already with the metagenomic data, with the microscopic pictures, with the pH changes, that it all supports. It's an ecosystem. And I hope you can continue doing that type of approach or that type of outlook with your samples that you still have. And I hope that as soon as possible, we can get new samples. Okay.
Yeah.
Okay.
We hope so too.
Okay.
Great.
Thank you both, Dalton and Jake and Beverly.
Thanks, Haida.
Jumping in here. And we will continue next week with a similar direction, the importance of microbes. And it will be Olivia Mason and Lauren Campbell from Florida State University.